Hello everyone, welcome to Sharda Campus YouTube channel. In today's video, we will learn uh, some fundamentals of the enzymes. So enzymes are basically biocatalysts that are produced by the human body, synthesized by the living cells. They are protein in the nature, colloidal and thermolabile means very sensitive to the temperature, and they are specific in nature. In the laboratory, the hydrolysis of the protein by the strong acid at the 100 degrees Celsius take a couple of the day, and the same protein is fully digested by the enzymes in the gastrointestinal tract at the body temperature, that is about 37 degrees Celsius, with a couple of the hours. So this is the role of the enzyme in the body, how it fasten up our biochemical reactions. There are certain classes of the enzyme, like first class is oxidoreductase. So it will work like catalyzes the oxidation reduction reactions. Second one is the transferase group of the enzyme that will catalyze the transfer of the functional group from the one molecule to the another molecule. Next one is hydrolysis. So it will catalyze the hydrolytic cleavage of the various compound. Lysis, so that will remove the group from the additional of the group to a double bond or other cleavage involving the electron rearrangement, additional or the removal of the water, ammonia and carbon dioxide, etc. Next class of the enzymes is isomerase group. So it will, as the name suggests, it will do only the intramolecular rearrangement. There will be no any changes in the molecular number or a molecular formula. Next one and last class of the enzyme is the ligase. So catalyze reaction in which two molecules are joined and ATP will used. There are certain factors that will affect on the enzymatic activity in the body. So like first one is concentration of the enzyme, concentration of the substrate, temperature effect, pH effect, product concentration effect, effect of the activators, effect of the time, effect of the light and radiation. So first one is the concentration of the enzyme. So how the concentration of the enzyme will affect on the enzymatic activity in the biochemical reactions. So as the concentration of the enzyme will increase, the velocity of the reaction that will be proportionality increase. So this factor is directly proportional to the velocity of the enzymatic reaction. Second one is the concentration of the substrate. So increase in the substrate concentration gradually increase the velocity of the enzyme reaction within the limited range of the substrate level. As we all know that for the enzymatic reaction, so there is a two primary requirement. First one is the enzyme and second one is the substrate. So this substrate will gradually increase. Meanwhile, the velocity of the enzymatic reaction will also increase. And the graph we can, as we can show in the figure, the shape of the graph we will obtain. Next one is the effect of the temperature. So as the temperature will increase up to the optimum temperature, the enzymatic reaction, the velocity of the enzymatic reaction will increase and up to the maximum level, and then that will decline. The bell-shaped curve that is usually observed, as we can show in the figure. Now, what is the reason behind the increasing the velocity? So, increase in the temperature result in the higher activation energy of the molecule and more molecule collision and interaction for the reaction to proceed faster. So this is the main reason why the rate of reaction is increased. So as the temperature will increase, the collision between the two molecules that will also increase, and that rate of reaction will proceed faster. The optimum temperature for the most of the enzyme is between 40 to 45 degrees Celsius. There is some exceptional enzymes like the venom phosphokinase, muscles adenylate kinase that will activate even at 100 degrees Celsius. In the general, when the enzymes are exposed to the temperature above the 50 degrees Celsius, the denaturation leading to the dearrangement in the native structure of the protein and the active site are seen. So we all know that enzymes are made up of the protein and they are thermolabile in the nature. So as the temperature will exceed about the 50 degrees Celsius, so its basic structure that it will lose. And for the primary requirement for the enzymatic reaction, that is a active site, so this active site will degrade and enzyme is not capable to do its work itself. Next factor is the effect of pH. So each enzyme has an optimum pH at which the velocity will be maximum. Below and above this pH, the enzymatic activity is much lower, and at extreme pH, the enzyme become total inactive. And the optimum